Okay, so welcome back to this next video in which we are discussing high-density lipoproteins and reverse cholesterol transport. Okay, right, so we now want to begin our discussion of the reverse cholesterol transport process. Okay, and the process is going to begin with the synthesis of HDL particles, because if we don't have HDL particles within the blood, we can't have reverse cholesterol transport. Reverse cholesterol transport, remember, is the process by which you take cholesterol from cholesterol-overloaded cells and return it back to the liver or also to steroidogenic cells within the adrenal glands, within the testes and the ovaries. Okay, right. So basically, let's start off with the discussion of how we produce HDL then. Okay, so it starts with the production of ApoA1, which is the core apolipoprotein within HDL. And this is done by both hepatocytes in the liver. So here is a picture of the liver. So we'll have a hepatocyte here. Okay, hugely enlarged so that we can actually draw stuff in it. And it's also synthesized actually by enterocytes, which are the cells which line the small intestine. Okay, so I might draw a portion of the duodenum here. Okay, so here's the C portion of the duodenum. And basically, the cells which line the small intestine, which we'll draw an example here, are called enterocytes. So entero means pertaining to the intestine, and cyte then means cell. Okay, similarly, cells within the liver are called hepatocytes. Hepato means pertaining to the liver, cyte means cell. So we've got a cell within the liver and a cell within the intestine, and these are both going to be responsible for secreting the starting components of high-density lipoproteins. Okay, so the first thing that they produce is the apolipoprotein A1. Okay, so here we have the production of apolipoprotein A1. Okay, right, and I think I'll pull one of these cells out and draw a bigger picture over here. So, let's have our cell here, like so, and it's now producing apolipoprotein A1. Okay, I'll draw this like so. Now, to make an HDL particle, what you need to do is give this apolipoprotein A1 some phospholipids at least, okay? So you need to give it some lecithin molecules to make the phospholipid monomer, okay? It doesn't hugely matter if you give it any cholesterol esters at this stage, okay? So instead of forming a complete HDL molecule which has the cholesterol esters, what we're aiming to produce is something which has an empty hydrophobic core. So let me draw this, okay? The starting thing that we're trying to produce here will look actually like this. Okay, it'll be in this disc shape because this hydrophobic core is empty because we haven't got any cholesterol esters in there yet. Okay, but it will have its apolipoprotein A1 here, and it will also have this phospholipid monomer that surrounds it, which will contain cholesterol molecules interdispersed. Okay, right. So, this structure that we're going to then create is what's known as a nascent discoid or discoidal, to describe the fact that its shape is in the shape of a disc. Okay, so nascent discoidal HDL. Nascent means newly created, discoidal means that it's in the shape of a disc, and then HDL just for high density lipoprotein. Okay, so this is what we're aiming to create. So what we need to give the apolipoprotein A1, therefore, is some lecithin molecules to make up the phospholipid monomer here, and also some cholesterol molecules to put into that phospholipid monomer. Okay, so this is going to happen at the hepatocyte or the enterocyte as well. Okay, so what the hepatocyte will have in its cell membrane is a transporter, okay? And this transporter is called the ABC transporter, ABC, and then A1. And I'll have a little bit of a discussion of ABC transporters in a moment. And this transporter is going to transport cholesterol and also phosphatidylcholine molecules across the membrane to the apolipoprotein A1, and that will form the nascent discoidal HDL in the blood, basically, and then the nascent discoidal HDL will then go into the blood and can then carry out reverse cholesterol transport. Okay, so what I now want to do a little bit of a discussion of is ABC transporters, because generally people have a very poor understanding of ABC transporters, and it's okay to have a very poor understanding of ABC transporters, because overall we don't have a very good understanding of ABC transporters. They aren't as heavily studied as they probably should be. Okay, so we're going to start off by looking at the structure of ABC transporters. We'll then look at all the different types of ABC transporter that are within the human body. Okay, we'll then discuss the mechanism of action of ABC transporters. Specifically, we'll look at the mechanism of a specific example of an ABC transporter which has been heavily studied, which is called the multi-drug resistance protein 1, also known as ABCB1. Okay, and then that will give us a good footing to understand what ABCA1 is going to be doing, and also we're going to see many other examples of transporters before this video is over, so it's a good thing to do, basically. So, we'll start off with the structure then of an ABC transporter. Now, this varies between different ABC transporters. Okay, so some ABC transporters are what are known as full transporters. And if you are a full transporter, then the normal structure for you to have is what I'm about to draw for you. Okay, and indeed ABCA1 is a full transporter, so the structure I'm about to draw is the case for ABCA1. However, some of the ones we'll see later will have a different structure. They'll be half transporters. Okay, and there are other ABC transporters which are full transporters which have a different structure to this. We're not going to see them in this video, but I will just give you their structure in a moment. Okay, for completeness. So here's the amino terminus of the polypeptide of the ABC transporter. It then has a membrane-spanning alpha helix, like so, and then 5 more membrane-spanning alpha helices, which are all clustered together. Okay, and this cluster of 6 membrane-spanning alpha helices together is known as Transmembrane Domain One, or for short, TMD1. So the T is for trans, the M is for membrane, the D is then for domain, and then of course 1 is just for 1. So TMD1 or Transmembrane Domain 1. So that's the name for the whole cluster of 6 membrane-spanning alpha helices. Then you have an important loop between Transmembrane Domain 1 and then Transmembrane Domain 2, and this contains a special domain which is on the cytoplasmic side of the cell membrane. Okay, and this special domain is called the Nucleotide Binding Domain 1, okay, or NBD1 for short. So NBD1 is short for nucleotide, that's the N, then the B is binding, and then the D is domain. So this is the Nucleotide Binding Domain 1. Now, nucleotide binding domains can also be known as ATP binding cassette, okay, which is the whole basis of these transporters being called ABC transporters. The ABC is short for ATP binding cassette, okay, and these nucleotide binding domains that we are going to see here, these can also be called the ATP binding cassettes. Okay, so we've only seen one nucleotide binding domain so far, so this is only the ATP binding cassette 1, okay, or you could abbreviate it to ABC1. So that's the basis of why these transporters are called the ABC transporters. It's for ATP binding cassette. Okay, then you go into transmembrane domain 2, which has again a cluster of 6 membrane-spanning alpha helices, as so. And then in the cytoplasmic carboxylic acid terminal of the polypeptide, you then have a second nucleotide binding domain here, and this is nucleotide binding domain 2, or you could call it the ATP binding cassette 2, the ABC2. Okay, so nucleotide binding domain 2, or if you like, ABC2. Okay, right, so this is the membrane spanning topology of the ABCA1 transporter, and many other ABC transporters also have this structure, and those will be called full ABC transporters. So you have 2 transmembrane domains, transmembrane domain 1 and transmembrane domain 2, and you also have these 2 nucleotide binding domains, which can also be called ATP binding cassettes, the nucleotide binding domain 1 and then also the nucleotide binding domain 2. Okay, now, not all ABC transporters are like this, though. Some of them are actually what are known as half transporters, but I think before we talk about half transporters, I'd just like to show you another type of full transporter that doesn't have the structure that I've just shown you there. Okay, so most full ABC transporters do have the structure that I have just shown you over the previous page, okay, and they are capable on their own of transporting molecules across the cell membrane, okay, but other full transporters have this structure instead. So let me show you another structure that full transporters can have. Okay, and an example of a ABC transporter, which is a full transporter that has this structure, is ABCC1. Okay, so you have the amino terminus this time extracellularly, and you then have a cluster of 5 membrane-spanning alpha helices, like so. Then you have a loop, and then you begin transmembrane domain 1. Okay, so this cluster of 5 membrane-spanning alpha helices here is often called transmembrane domain 0. Okay, so this is the additional bit that you have in this different type of full transporter. Then it's just the same. You have transmembrane domain 1, you then have nucleotide binding domain 1, also called the ATP binding cassette 1. You then have the transmembrane domain 2 over here, with its 6 membrane-spanning alpha helices, like so. And then on the cytoplasmic side, you then have transmembrane domain 2, which then has a carboxylic acid tail here. Okay, so this is the nucleotide binding domain 2 here, or the ATP binding cassette 2. Okay, so certain full transporters have this additional little bit here, known as transmembrane domain 0, and an example of that would be ABCC1. Okay, but we won't see ABC members at all in this video. Okay, that's just there for completeness. Okay, and these 2 are full transporters, i.e. if you give them ATP, they will transport proteins across, or, well, substances, solute substances, not necessarily just proteins. Lipids, obviously, is the example that we're interested in. They will transport their solute across the cell membrane, basically. Okay, now all ABC transporters are actually primary active transporters. They require ATP in order to transport their solute across the cell membrane. Okay, right. Now let me discuss these transporters which are half transporters, and we will see examples of half transporters within this video. Okay, so half transporters are not capable of actually transporting the solute across the cell membrane on their own. Instead, they have to dimerize with another half transporter, which is often an identical half transporter to the first one. Okay, so often it will homodimerize with another half transporter, and then it will form a functional transporter which can transport solute molecules across the cell membrane. Okay, so let me show you the membrane-spanning topology of these half transporters. So, the amino terminus will be on the cytoplasmic side, and then you'll have a single nucleotide binding domain in these half transporters. Okay, so you have a single nucleotide binding domain, which is on the amino side this time, and then you'll have a single transmembrane domain, which will consist of 6 membrane-spanning alpha helices, and then the carboxylic acid tail. So hopefully you can see that this is kind of half of a full transporter. A full transporter needs 2 nucleotide binding domains and 2 transmembrane domains, whereas a half transporter only has a single nucleotide binding domain and a single transmembrane domain. Okay, so to make the full functional transporter, it has to dimerize with another half transporter so that they can then together form a functional transporter, which will have 2 transmembrane domains and 2 nucleotide binding domains. Okay, now these dimers that they form are often homodimers. Okay, and what that means is homo means the same, so it means that the other half transporter that it has dimerized with is basically exactly the same as itself. Okay, so it didn't go off the rails and pick a different type of half transporter, ABC transporter. Okay, but you can find heterodimers where different types of ABC transporters, which are both half transporters, have linked together to make a chimera effectively. Okay, right, so let me now tell you about the different ABC transporter proteins that humans have. Okay, so there are 49 ABC transporter proteins within the human genome. Some of them will be full transporters, which will function on their own. Some of them will be half transporters, which therefore need to dimerize to form a functional transporter. Okay, so there are 49 ABCs, and some of them will be these full transporters which have this transmembrane domain 0. Okay, so to understand the 49 different ABCs, what we do is we divide them into 7 families. Okay, so the first family is the ABCA family. Okay, and this contains 12 members, and their naming is very, very sensible apart from the fact that it has 1 blip in it. Okay, so you have ABCA1 is a member of this first family. You then have ABCA2, ABCA3, and then it goes on ABCA4, ABCA5, ABCA6, ABCA7, ABCA8, ABCA9. Then we get to ABCA10, and I should have put ... there. Okay, so we get to ABCA10, and then there is no ABCA11. Okay, so you skip ABCA11, there is no ABCA11. Then you have ABCA12, and then finally ABCA13, to give you a total of 12 different ABCA proteins. Now, all of the ABCA proteins, of which ABCA1 is the one we're very interested in, because that is the one which is on hepatocytes and it's on enterocytes, and it's going to be transporting cholesterol molecules and also lecithin molecules over to the apolipoprotein A1. Okay, all of these are full transporters. Okay, right, so they have the same structure as I showed you originally. Okay, they have this structure. They have the nice standard structure where we have 2 transmembrane domains. We don't have that silly transmembrane domain 0, and then we have 2 nucleotide binding domains. Okay, right. Let's go on to the second family. The second family is ABCBs. Okay, and this family contains 11 members, and the naming is perfectly sensible. So you have ABCB1, ABCB2, and then all the way up to ABCB11. Okay, and we will see members of the ABCB family in this video. Okay, which will be which will be relevant to the reverse cholesterol transport pathway. Okay, right. We're also going to be seeing ABCB1 in a moment, just to look at the mechanism by which these things work, even though that ABCB1 isn't terribly relevant to the reverse cholesterol transport process. Okay, right. Now the ABCC family. Okay, and remember ABCC1 was this transporter which had this transmembrane domain 0 here, but it was a full transporter. Okay, the ABCC family has 13 members, and again the naming is perfect. So it's just ABCC1, ABCC2, ABCC3, all the way up to ABCC13. Okay, so that's fine. Then we've got the ABCD family, which is the nicely named one, okay, if you appreciate such silly jokes. Then this family has 4 members within it, okay, so you have ABCD1, ABCD2, ABCD3, and ABCD4, okay, and we're not going to see any of those in this video. Then we've got ABCE family, and ABCE family is the unlucky one. It's only got 1 member, okay. Then we go to the ABCF family. This contains 3 members, so you have ABCF1, ABCF2, and ABCF3. And then finally the ABCG family, which is the least well named, okay, so there's holes all over the place in ABCG. Okay, so you have ABCG1, you then have ABCG2, you don't have an ABCG3, so you go straight on to ABCG4, you then have an ABCG5, then you don't have ABCG6 or ABCG7, but you do then have ABCG8. And that now completes the list of the names, at least, of all human ABC proteins that are currently known. Okay, so let's just check that I have listed out 49 of the things. So we'll start with the ABCA family. We've got 12 members there. Okay, even though it goes up to 13, remember there is no 11. Okay, so that's 12. ABCB family, we've got 11. That takes us up to 23. The ABCC family, we've got 13. That takes us up to 36. The ABCD family, we've got 4, that takes us up to 40. The ABCE family, we've got 1, that takes us up to 41. ABCF family, we've got 3, that takes us up to 44. And then ABCG family, we've got 5, that takes us up to 49, as I said. Okay, right, so that's the list of all the names of the ABC proteins within humans. Okay, what I now want to talk to you about is the mechanism of action of an ABC transporter. Okay, and there is one ABC transporter that has been very heavily studied. Okay, and this is the lucky ABCB1. Okay, and it's so important that it's also got other names. So it's this first member of the ABCB family. It's a full transporter, and it's got the same structure as we saw originally. So it's got this nice structure here, the same as ABCA1. Okay, right, and it's so important that it's got other names as well. So it's also called P-glycoprotein. Okay, and for short, P-glycoprotein is often abbreviated to P, and then you put lowercase g, lowercase p for glycoprotein. Okay, but it's got another name as well in addition to P-glycoprotein. It's also called the multi-drug resistance protein, and that's the multi-drug resistance protein 1. And for short, the multi-drug resistance protein 1 is often abbreviated to MDR1. Okay, so M is for multi, D is for drug, R is for resistance, and then we've got 1. So that's MDR1. Okay, now, why is it called that? Well, basically, MDR1 has been found to be important in drug resistance within certain types of cancer cells. Okay, specifically, it's been found to be important within drug-resistant liver cancer, drug-resistant kidney cancer, and also drug-resistant colon cancer. Okay, so if your cancer cells overexpress this protein, the ABCB1 or the multi-drug resistant protein 1, it's not a good sign, because basically this is a transporter and it transports things out of the cytoplasm of the cell. Okay, so if I draw a little picture here, let's say this is our liver, kidney or colon cancer cell. Basically, here is the multi-drug resistant protein 1, the MDR1 protein, and basically it can transport your drug molecules out of the cells. Now, transporters which export molecules, i.e. they transport them out, are often called exporters. So this is an exporter. Okay, transporters which bring things into the cells are often called importers. So that's just a little bit of nomenclature. So this drug resistance protein 1 is extremely good at latching onto anti-cancer chemotherapy drugs and chucking them out of the cell, basically. And that's a running theme for these ABC transporters, that they're actually capable of transporting many different ligands. Okay, so it's not just the case that they have one molecule which they like to transport across the cell membrane, and that's the only molecule they will transport across the cell membrane. Often they have multiple different molecules which they can transport across the cell membrane, and this MDR1 is a good example. Okay, so it transports all sorts of drugs. So some famous anti-cancer chemotherapies that it's capable of transporting include vinblastine and also the truly horrible drug doxorubicin. Vinblastine is an antimitotic. Doxorubicin is a DNA intercalator, a truly foul drug. Okay, and basically, if you overexpress MDR1 on your surface, you are invulnerable to these drugs because you'll just get them pumped out, basically. And drug-resistant liver, kidney, and colon cancer cells have been found to do this. Okay, so you might ask, well, how have they done this? Well, it won't have been the case that before you started treatment, the cancer cells were resistant. But what you have to understand about cancer is that you have this massive great tumour, which consists of a huge number of cells which have all got genetic instability. Okay, so they have mutations in the mechanisms which prevent you suffering mutations and continuing with those mutations. Okay, so most cells, if they get a mutation in the DNA, they will activate all sorts of repair mechanisms to repair it, and if they can't repair it, they'll then commit suicide. Cancer cells don't do this, basically. They allow the mutation to stay, and that's what we mean by genetic instability. You're getting this pool of cells that are all undergoing mutations at a ridiculous rate. So you get loads of cells within this one tumour which are all different. So let me draw a tumour here. Basically, loads of the cells in here will all be genetically different, so they'll all have very different characteristics. Now, some of them will suffer mutations that actually kill them. So, for instance, if they get a mutation in one of the essential complexes in the electron transport chain of respiration, then that will kill them. Okay, but some of them might just undergo mutations that mean that they overexpress MDR1, so maybe a mutation in the promoter region for MDR1 which causes MDR1 to be completely overexpressed. Okay, and then when you start firing these horrible drugs at the cancer cells, so for instance vinblastine or doxorubicin, when you give the patient these drugs, of course, it will kill all of the cells which don't overexpress MDR1, but there will be one cell, most likely, in there that has just happened to undergo the mutation that means that it's got far too much MDR1 on its surface, and now it will survive. So it's just a natural selection process here. And then all of its friends will die, and then it will go, whippy, I've got more space now. It will divide and produce a new tumour which consists of cells which have all got this same mutation that it had, and therefore have all got drug resistance to these drugs. Okay, so that's how you end up with tumours which are completely resistant to the drugs. Okay, you started off with just maybe a few cells which were resistant. Those were the only ones that survived. You set up a selection pressure when you gave the drug, and they've now repopulated the tumour, and the tumour has therefore evolved. The whole population of cells has evolved. Okay, right. So That's why MDR1 has been so heavily studied, because it is overexpressed within drug-resistant liver, kidney, and colon cells. Okay, so obviously there has been thought as to whether it would be good to create a pharmacological blocker of this MDR1 channel, which would therefore stop the drug resistance in these cells. Now, from what I've told you, that would seem a fantastic idea, but you're only seeing the involvement of ABCB1 in pathology. Okay, you should always ask, what is the role of this in physiology? Where else is this channel used in physiology? Remember the COX-2 selective drugs. We thought they were going to be fantastic because we thought COX-2 was this evil enzyme which became active during inflammation. Okay, and which had no physiological role. Okay, so we created COX-2 selective blockers to take out this evil enzyme and not take out COX-1, which was the good enzyme which had physiological function. Okay, and of course the COX-2 inhibitors, characterized by rofecoxib, then caused heart attack in the people who took them. Okay, and rofecoxib was withdrawn in 2004, and I think it caused, caused huge damage to the drugs company that created it, and also obviously the victims who took it. Okay, so what's the physiological function of ABCB1? Well, you find that it's actually on the blood-brain barrier, and it's protecting your brain against horrible drugs in your blood, basically. It pumps all of the horrible drugs back into the blood, basically, and away from the brain. So blocking it might not be such a good idea. Okay, right. However, it has been intensively studied because of its involvement in drug resistance within liver, kidney, and colon cancer, and therefore a mechanism for it is to some extent understood. Okay, so I'm now going to present this mechanism so that we've got some sort of groundwork to understand how all the ABC transporters work, because although we don't know that this mechanism extends to all the other ABC transporters, it would probably be naive in the least to say that it does, most definitely. But it probably is similar, so it will give us at least some intuition for what these things are doing. Okay, right. So, firstly, this mechanism, you will not find it in the textbook, at least I haven't found it in the textbook. So once again, I'll give you the reference where I got this from. So I got it from a review written by Linton in 2007. Okay, so if you search that on Google, you'll probably be able to get the same thing up. Okay, right. So, this is what's known as the ATP Switch Model. Okay, right. And I think we will have a break before we discuss this, and we'll start the discussion of this ATP switch model by which the multi-drug resistance protein 1 actually transports drug molecules out of the cell in the next video.